In this video, we're going to cover the cerebral cortex and its organization. Its organization in terms of the layers and the basic functional units being the cortical columns and kind of what kind of cells we'll find in the cortex. And then also the organization of our sensory and motor systems. And what we're going to find here is that we're going to be building on more basic bits of information in order to come up with more complex perceptions. And whenever we have motor output, we kind of start with a bigger idea, the idea of I want to reach out and grab a thing, and then this goes, and we go from a big idea to much smaller ideas. What do I need to do with my shoulder, my elbow, my hand, right? So for sensory systems, we're sort of building bigger ideas from smaller ones. And in motor systems, we're breaking bigger ideas down into smaller, more manageable ones as we move from association to primary motor cortex. Because the whole goal of the brain there is to regulate our behavior and help us make sense of the world. So how does this feller here know he's looking at a dog? You know, he kind of takes into account the height of it, the number of legs, and the shape of the face. And there's certain neurons that are more responsive to, well, dogs, and some are more responsive to cats because they've learned, well, if I see kind of a long snout and it's got this collar on it and there's a human behind it and it's got pointy ears and, oh, it's just kind of adorable looking, well, that's a dog, right? We create that conscious perception by building it bitwise. Now, where the magic happens in the cortex is, of course, in the layers with all the different cells that are found in them. And in our cerebral cortex, the organization is partly vertical, that is between different layers. So we have 6 layers in almost all of our cerebral cortex. Some is, missing a layer here or there. And then we take these different layers, and depending on where you move around in the cortex, from the front to the back, for example, we then get different functions, but we don't really see different organization. No matter where you are in the cortex, you got pretty much the same cells, the same number of layers, the same wiring. So we have these basic functional units that we call cortical columns, and the only thing that differs between them is who are they getting input from and who are they talking to. But no matter where you are, it's the same 6 layers. And we see pretty much the same pattern of organization. Our input from the thalamus is going to arrive at layer 4. And that's going to get spread to a few different kinds of projection neurons. Some are up here in layer 2. They'll go to other cortical areas. So will layer 3. So 2 and 3 we often lump together. We just call it layer 2, 3. Sometimes we'll separate them. Cells are a little smaller in 2 than they are in 3, and the cells in 3 can, cross to the other side of the brain. So 4 is our input, and we spread it up to 2 and 3 so the cortex can talk to itself, right? We can build on small ideas to make bigger ideas. We can turn letters into words and words into paragraphs and stuff like that. We can also spread on down to layers 5 and 6, and these are going to project out of the cortex. Now notice, these are the 2 layers on the bottom. So if you wanted to project out of the cortex, it would make sense to have the bottom 2 layers do that because they're closer to the bottom, which is where you want to project. When we talk about projections out of the cortex, for the most part, we're talking about layer 5. And layer 5 has the absolute biggest neurons here. You can see they're pretty darn big over here. This is my association cortex in the frontal lobes. And over here in the motor cortex, notice what happens. Yeah, we've got the same 6 layers as our frontal cortex here. But what do we have? A whole lot more layer 5. And that's because these are my upper motor neurons. These are the ones that are going to project on down and talk to all those lower motor neurons so I can move. And so it makes sense for the primary motor cortex to have a bigger layer 5. It doesn't have a very big layer 4. I'm not so much worried about what the thalamus is telling me here, right? That's going to be more for sensory areas. So we'll have the same layers, but the size of them varies What doesn't really vary as much is the connectivity and the types of cells that we find there. So in layer 1, if you prefer a table, here it is, no cells. It's just a bunch of dendrites and axons. We'll get that matrix input from the thalamus. Other cortical inputs can arrive here too. Layers 2 and 3, these have small and large neurons respectively, and that's just cortex talking to cortex. Layer 4 is where the thalamic input arrives and then spreads throughout these layers, and then 5 and 6 would be my outputs. 5 projects to all those subcortical structures except the thalamus. That's the job of layer 6. Now, all of those layers, those 6 layers are here, and what we find is all the neurons that live kind of near one another all respond to the same basic part of your body or the world. They have, in other words, the same receptive field. So this is easier to think about with the visual system, and that's what we're looking at here. So all those neurons that live near one another are within what we call a cortical column. In other words, it's a column of cells, right? So layers 1 through 6. This column all responds to the same little area of your retina, for example, if we're in the primary visual cortex. Now, that doesn't mean they respond to the same thing going on in that little spot of your visual field. Some neurons are responsive when there's a line and it faces a certain orientation. And if you move to their neighbor, well, they like a slightly different orientation. Notice we go from kind of vertical here to horizontal and back. So we have neurons that are responsive to all angles of lines within this very small part of your visual field. And then there are neurons that respond to all the different possible colors. And so based on the activity of neurons in this cortical column, it can encode what was the angle of the line there? What was the rough color? Was it more blue or green or red? What was going on in my different eyes? Right? I have my ocular dominance column still. Did my left eye agree with my right eye? If so, hey, it's probably in focus. If they're different, well, this is probably behind or in front of what I'm looking at. And so by having all these neurons in this cortical column respond to all the different possible values for what we could see, or if it's in the somatosensory cortex, what we could feel. The pattern of activity across all these columns will then let us reconstruct our entire visual field or our entire body, right? Because different columns are going to talk to each other. Remember layers 2 and 3? That's cortex talking to cortex. And so we can hear what the neighbors are doing. Now, within each cortical column, there's 2 types of cells broadly. There's the excitatory and inhibitory neurons. And for each of those, we can dichotomize them again. So for excitatory neurons, there's 2 types. There's the pyramidal neurons. They have these kind of triangular bodies here, that cell body that looks a little bit like a pyramid. Pyramidal neurons tend to project. And then we have stellate neurons. This kind of looks like a star. That's why it's called stellate, and they stay local. So there's an order of operations here. That thalamic input, notice that's what's coming in here, and it arrives at layer 4 to a stellate neuron. And that stellate neuron spreads the excitation onto a pyramidal neuron. And that pyramidal neuron then projects out to do a thing. Maybe go tell the basal ganglia something, or maybe go tell a lower motor neuron something, or whatever it may be. But that's our order of operations. We get some input onto a stellate cell, it spreads to a pyramidal cell, and then that goes and tells somebody about it. Depending on the types of input we get, that'll determine the pattern of pyramidal cell activity and thus the type of output from that cortical column to say whether or not you're looking at something red or blue or green or vertical or horizontal or 30 degrees or whatever it may be. Now, of course, we also have the inhibitory neurons, and this is incredibly diverse, and we're going to just think of them again dichotomously. What's their function? Now, some of them are there for good old lateral inhibition. And this is the same thing it's been in every other lecture. This is to focus activity across space. So if I'm active My neighbors shouldn't be, unless they're also getting input. Okay, so here's a basic schematic. If this pyramidal cell's active, it turns on an inhibitory interneuron that turns off all the neighbors, right? So if this is the one active cell, this SST is just it's just for somatostatin. Don't worry about that. That absolutely doesn't matter. It's an inhibitory neuron. That's all that matters. And so it inhibits all the neighbors. And so, hey, only these strongly excited neurons are active. Okay, without lateral inhibition, what might this look like? So this is a sensory system out there in the skin. Same thing in the cortex. If we have some stimulus arriving here, it could be pressure on the skin, could be excitatory input in the cortex, whatever it may be, one neuron is really, really active, but so are some of the neighbors, right? A little less active. Where was that stimulus? Where did it arrive? Well, if we go down here to our next neurons in the relay, they have a hard time figuring it out. Yeah, I'm a lot more active because I got a lot of my input from that really active neuron, but hey, axon projections ain't perfect. Look, this stimulated neuron is talking to my neighbors too, a little bit, not as much, but a little bit. And so now all 5 of these neurons are stimulated by these original 3. And so over here, graphically, that stimulus produced a lot of activity in one place and a little bit in the neighbors. But if we go down to the next set of neurons, sloppy axonal projections are going to spread that excitation out. That's not what we want. We want to focus it. And that's where lateral inhibition comes in. Notice all we're doing here is adding some inhibitory interneurons. So it's the same stimulus. I'm still mostly stimulating this neuron and a little bit of the neighbors. But what happens? I go down here, I strongly excite this neuron, and I'm kind of weakly exciting its neighbors, and that neuron turns on these inhibitory neurons. Sure, it gets inhibited a little bit, but it's also excited a lot. And so it's still activated. The neighbors that are weakly excited, they're also getting inhibited. So that cancels it out. And what do we end up with? We're not widening out that area of activity. We're focusing it. So now I'm more active if I got input from the stimulated neuron. And if I didn't, I'm less active. So notice this is focusing the activity across space. So how can I localize where a thing is? Well, since the stimulus was here, this is the neuron that was active, not the neighbors. And that's through lateral inhibition. Feedforward inhibition is a new one, but same idea. We're just going to work across time. Now remember, neurons that fire together, wire together, right? And if I'm a neuron that responds to dogs, I'm looking for a snout and pointy ears, and I want to make sure I'm seeing those 2 things at the same time. Here's where feed-forward inhibition comes in. So we'll start on the bottom here. And we've got 3 possible thalamic inputs. We'll call them 1, 2, and 3. I didn't break the bank on that creativity there, but again, 1, 2, and 3. And the same up here, 1, 2, and 3. So 3 different thalamic inputs. Now notice what's happening on the bottom here. Only one of them is providing input, and we're going to see 2 possible things. So here's my thalamic input. Now just that excitatory input here. All right, let's say I spread out a little glutamate, and I kind of excite this pyramidal neuron just a little bit. Just a little excitation, and that's what's shown here. This is what it would look like if we just got excitatory input, but that's not all that's happening. I'm also exciting this inhibitory neuron here. Don't worry about what that's parvalbumin, doesn't matter. It's just an inhibitory interneuron. So you can replace that with just IN for inhibitory neuron. Okay, I'm also exciting this one. Okay, so I get excitation, and it's almost immediately followed up with inhibition. Okay, how does that affect the excitation? Notice what's happened here. I've lost a lot of that inhibition over here. So without the feed-forward inhibition I'm getting over here from this neuron, I would be excited. I'd be depolarized for much longer than what we actually see in cortical neurons. Notice there's a very brief period of time where I'm depolarized, and then after that, I'm quiet. So if I need to see that it has pointy ears and a snout and a collar on. Those are my 3 thalamic inputs. I only see one of those. I see it's got a snout. Okay, I'm not thinking it's a dog. Right, that's only one piece of input. Doesn't have the pointy ears. No collar. All right, so I'm not getting up to threshold. Now, what if I see one animal with a snout, another animal with a collar, and another animal with pointy ears? I'm getting the 3 inputs, but they're not at the same time because they're from different animals. I'm looking at a cat and a pig and, I don't know, a human with a collar on. I don't know. But there's my 3 inputs, but they're not coming at the same time. I've seen the 3 things I want, but at different times. It's not a dog. Okay, so I'm getting, again, very brief input. These 3 thalamic neurons aren't firing together, so I'm not really looking at a dog. I'm looking at a cat and a pig and a human with a collar on. Well, that's not what's going to turn on this dog-responsive neuron, and it won't because of feed-forward inhibition. Notice I get some depolarization, but I pretty quickly go back to rest. And I get a little depolarization, and I pretty quickly go back down, and a little, and I go back down. I don't keep that depolarization around. It can't build on the old depolarization because of this feed-forward inhibition. Okay, so what if I am looking at a dog? I see, hey, it's got a snout and pointy ears, and look at that, it's got a collar on, and I noticed it just a little bit later, but I still noticed it. I'm seeing it at pretty much the same time. What happens? Well, now, even though I get very brief excitation, they're happening at the same time, and what happens? I hit threshold. And what's going on here? There's a spike. They just cut it off. There's an action potential. I fired. I saw a dog. And that's what my perception is going to be. Hey, that thing must be a dog because I've got this coincident thalamic input happening. And I know it happened at the same time because feed-forward inhibition prevents me from caring about old stuff. Okay, so we've got lateral inhibition to make sure only neurons that are getting input are excited. And then we've got feed-forward inhibition to make sure that only input that arrives together can pile up and excite that cortical neuron up to threshold. So I've done some kind of a calculation in my cortical column. How do I go tell somebody about it? And that's through these things called mostly association fibers. Association fibers are ipsilateral connections, that is on the same side. So I'm going to go talk to my neighbors. We got local connections. Hey, I'm just going to talk to, the gyrus right next door. We got neighborhood connections. That means, we're near one another, but I'm going to maybe go a little further away. And then we've got long association fibers. I'm going from the frontal lobes to the occipital lobes and vice versa. No, you don't have to worry about distinguishing these. Just know that we have association fibers from layers 2 and 3 that can connect very close or very distant structures in the cortex. And yes, it may be, within the same area or between lobes. Then we have connections that cross the midline. We've got to talk to the other side of the brain, and for the most part, that's going to be in this corpus callosum right here. These are going to be my commisural or my contralateral connections. Now that corpus callosum, it's got a front, it's got a back, and there's an organization. And sure, people name it. There's a rostrum, a genu, a body, and a splenium. Don't sweat that. But do know that if you're at the front of the corpus callosum, you're getting input from the front of the brain. And if you're kind of in the middle, you're getting the middle of the brain, more parietal lobe stuff. And if you're at the back, you get the occipital lobes. There's an organization, and it makes sense. Key thing here is that the corpus callosum allows the 2 sides of the brain to talk to each other. So we get our contralateral connections. Okay, so here, make sure we get an idea of inputs are arriving from the thalamus in layers, mostly 4, but a little bit of 1. And then that gets spread within the column. Layers 2 and 3 provide output to other cortical areas. Layers 5 and 6 talk to non-cortical areas, with layer 6 talking to the thalamus, layer 5 talking to everything else. Pyramidal neurons are the ones that project, and the stellate ones tend to stay local. Lateral inhibition focuses across space, so only neurons that are receiving strong excitatory input are active, and their neighbors get turned off. Feed-forward inhibition makes sure that cortical neurons only think that two things are happening together if they really do. We go from this kind of longer excitatory postsynaptic potential where we don't know. There's no difference in the amount of depolarization, whether it's immediate or after some delay. We're going to go from that to this much more rapid EPSP, where now I'm only really excited immediately by that input. And then it's quickly lost. So we know that if neurons are firing together, it's because something actually happened at the same time. Okay, so let's go through our sensory and motor systems here. And really it's going to be just stepwise processing. For sensory systems, we tend to build in complexity. We look at the angles of lines, we look at the amount of red, green, and blue, and we can determine objects. And then we can look at our memories of those objects and put a name to them. And maybe we have experience with them. We can put some emotions to them. We're going to go from very simple ideas, just angles of lines, to much more complex ideas. With motor systems, it's kind of the opposite. We have a big idea of a grand action we want to do, I want to juggle, and we break that down into much simpler movements that get carried out by upper and lower motor neurons. It's good to have some idea of the map of the cortex. We'll review this in Lab 9, but it's good to know where the primary sensory regions are, and there's an organization that makes sense, right? Temporal lobes, these are getting input from that medial geniculate nucleus. So hey, over here, this is auditory cortex. There's the primary right there. Primary visual cortex back in the occipital lobes. Primary somatosensory, right? Olfactory over here. Good to know where these things are. The insula, taste, right? Balance is buried somewhere in there. If you prefer tables, there you go. We've gone through all of these in their own respective lectures. Just know we got a spot for things. And not just one, but a couple. So there's what we call a primary cortex, and that's going to deal with really only that thing, just vision. So the primary visual cortex only deals with visual input, and it's very nitty-gritty, itsy-bitsy details. The association, though, the visual association cortices take that information from the primary visual cortex and start to figure out, well, what the heck is it? Where is it? I'm going to integrate or associate both visual input and somatosensory input. Where is this thing relative to me? Or visual input with language. What's the name of that thing? So in primary, we deal with only one thing, and it's just the details. We make perceptions, complicated perceptions, by associating multiple types of information in association cortices. And what I'd like you to notice, I'm going to go back a slide, is that the association cortices are like right outside of the visual cortex. And what we'll see with vision, really with any of these, but vision's a great example, as we move closer to, let's say, the somatosensory cortex here. When we go up here into this association area, here's where we say where a thing is. So I've got visual input and somatosensory input. I know where I am. I know where an object is in my visual field. Based on that and my head position and all that stuff, I can figure out where is it in the world. I'm kind of averaging the functions of these two primary cortices. What about naming a thing? Well, sounds, names, that's going to be over here, temporal lobe stuff. Memory, of course, medial temporal lobes. So I'm not going to go up. I'm going to go down into this more ventral association cortex, and I'm going to take my visual information and I'll integrate it with sounds and memories, and now I can name the object. So think of association as kind of averaging the function of different primary cortices. That is an oversimplification, but I think it'll work for you. Okay, so let's start off with vision because this is easy to visualize. No surprise there. Okay, primary visual cortex, V1, vision 1, primary. And in the primary visual cortex, those neurons respond to incredibly basic types of information. What's the angle of a line? Does it seem to be moving perhaps? Is there retinal disparity? What's the color, red versus green versus blue? And then we're going to go to association cortices to figure out more complicated stuff, like what is it and where is it. So if we were to record across different neurons, now we're moving kind of around in a column. All right, so they're going to put a lesion in here, and what they're doing is inserting an electrode, and they're just recording along the way. So here they're recording as they move the electrode along. So as we're moving distance, what we're doing is listening to different neurons in the column. And what you'll notice, some of them respond to negative 60 degrees. And as we move to the neighbors, we're getting closer to 0 degrees. That is a vertical line. And we keep moving and we keep rotating clockwise here. Now we're responsive to 30. Oh, positive 60. Now a flat line. And hey, now we're negative again. We've kind of gone through the whole column. And now we're into a neighboring column. And what is it doing? Well, we're going through 360 degrees yet again. So neighboring neurons are going to respond to similar but slightly different information. Yes, we're still telling you the angle of the line. Depending on which neuron we listen to, we're going to perceive a different angle for that line. So within that cortical column, we've got a neuron for every possible angle and every possible color. Now, how are we sensitive to lines? Well, that's because most of our neurons in the primary visual cortex are orientation selective. So here's an example. We're recording from one neuron and we're showing it different lines. A flat line, tilted 15 or 45 or whatever it is, right? We're adding tilt to the line, and what I like most is vertical lines. Ooh, love a vertical line. Hey, that's not as vertical. Yeah, it's not vertical. That's not vertical. This is what gets me going. Right in here. Don't show me a horizontal. No, I like the vertical line. I sort of respond when lines are kind of vertical, but I give you the most response when I get a vertical line. Now, the receptive field can be either simple, complex, or hypercomplex here in the primary visual cortex. So a simple cell, I'm turned on when there's a line right here in the middle, but if you move it to the surrounding areas, I get inhibited. And we'll see why this is simple in just a little bit. Then I could be a little more complicated, what we call a complex cell. I've got my stimulatory region, but there's no inhibitory region. You can move this line around. As long as it's the right angle, I'm happy. I don't care if it's in the dead center of my receptive field. It could be a little up, a little down. Heck, it could even be a white line instead of a black line. I'm still fine with that. And then there are what we call hypercomplex cells. I care when there's a line and it ends. So I need to see a line of a certain angle, but it has to stop. If it keeps going, no thank you. I'm not as excited about that. So I'm here to tell you when there's a line of a certain angle that ends. Okay, how can we possibly get these crazy receptive fields? It's just stepwise visual processing. Okay, so in layer 4, that's where we're getting input from the thalamus, which is where we got input from retinal ganglion cells. And if you remember retinal ganglion cells, and I'm sure you do, they have an center surround receptive field. So what if I get input from 4 different retinal ganglion cells via, of course, these layer 4 neurons, and their receptive fields are just a little off of each other? What's going to get me maximally excited is if I can get some light that turns on these 4 receptive fields, and that would need to be a line, not a dot. When there's a line there, now I'm going to be super duper excited because I'm getting excitatory input from all four of my inputs. And so what does the receptive field for this neuron look like? A simple receptive field here where I have an excitatory region and an inhibitory region. If I put that line of light over here, all I'm doing is stimulating the surround. And therefore, I'm going to see a whole lot less activity in that neuron. And so, if I get the angle of the light just right, I'm totally excited. That's what's going on here. Now notice as I deviate from that angle, I'm including less of my on and more of my off. I'm not quite as excited. Yes, part of this excites me. This part's all good, but this part, not. And that's how we get orientation selectivity. Okay, what about a complex cell? Again, it's stepwise building. Now let's take a few simple cells. They all respond to the same orientation, in this case vertical, but some of them are on in the center and some are off in the center. Okay, so if I throw a line in the center, I'm going to turn on two of my sources, but not the other. I'll still get excited. What if I put the light over here? I'll turn off some of my sources, yes, but I'll turn on others. And again, I'll get excitatory input. So I don't care where the line is, as long as it's vertical, this complex cell is happy. Now, this is not unique to vision. This is kind of the rule for sensory systems. We start with simple information, in this case, points of light. We turn that into slightly more complex stuff, lines. Then guess what we do with lines? We turn lines into shapes. What if I have 4 lines? Well, I'm going to turn on, right, one of these simple cells, another simple cell, a neighboring simple cell. I get these 4 simple cells active that are near one another, and now I've turned 4 lines into one shape. Now I've got myself, I'm going to talk to a square neuron or a diamond neuron, whatever you want to call it. So it's just building on simple information. And that is the case for pretty much all of our sensory stuff. So for vision, what do? Well, primary visual cortex. What's the angle of the line? Is there an edge? Right? Is one of those hyper-complex cells happy about it? Same thing for color opponency. That's still going on. Is it more red, green, blue? Is it the same? You tell me. And who are we going to tell? The next visual cortex, V2. V2 comes after V1. Hey, cool. That makes sense. I put some lines together. Next thing you know, over here, whoa, I got a shape. Or if I know it's a lot of red, a lot of green, and no blue, hey, that's yellow. I've finally figured the color out. Okay, where do I go from here? Well, it depends. What are you trying to do? You want to figure out what it is? Head on over to V4. What are all the shapes and colors that I see? I can tell you what the object is. I think it's a stop sign. Oh, well, what do I do with that? Okay, so now we move outside of the visual stuff and into one of those association cortices in the temporal lobes. And I know it's a stop sign, and I know that because that object I've heard of before. I associate it with language. Great. If it's a face, do I recognize the face? Or if it's an object that I really care about? You know, I know the difference between some really specialized tools or musical instruments or cars or whatever. I go outside of the visual cortex into the temporal lobes and see, do I have a memory of this thing? And I'll tell you what it is. Well, maybe we care where it is. That's going to be the green stuff here. We go from V1 and V2 on to V3. Okay, I see your shape. That's excellent. Is it moving? Does it seem like it's in a different place than it was a little while ago? If so, I'll tell you it is. We've got motion selective neurons. Okay, so maybe it is moving. Let's go on to V5. Which direction? Okay, so I know where it is and whether it's moving, and now let's keep on moving. We're not going to the temporal lobes, we're going to the parietal lobes to say, where is it? Now let's walk through these two streams, give them their own little summary. So what is it? The ventral stream. We're heading down into the temporal lobes. Again, we're building on old information. Okay, there's some Brodmann area numbers here. You don't really need to worry about those. That's just in case you care. So anyway, as we leave, we go into these now association visual areas here. What is it? So we go into this V4. We got some color neurons that'll tell you what color it is. We got neurons that'll tell you if it's a face. We got neurons that'll tell you what the shape is, what form is it. Right, there's an organization from medial to lateral. That's summarized up here. Okay, we figured that stuff out. Now let's go over into the temporal cortex and say, well, what the heck is it? And here again, we're averaging function. So I know the general object. Let me go here, which is kind of close to, oh, hey, look at this, the entorhinal cortex. And you know what's coming, memory. Oh, the amygdala's over here, emotion. Do I recognize that thing, and have I interacted with it before? Do I have a feeling about it? So do I have a memory and a feeling about it? That's what's going to go on here. Here it is in kind of flow chart version. We got amygdala, hippocampus. And notice my visual pathway, the ventral stream, is going down into the temporal lobes, where we have a whole bunch of memory and emotion stuff going on. And so I can name it. The dorsal stream, we don't need to go that way. We need to go up to where we have more spatial stuff. So we're going to go a different way. We're going to say, hey, is there motion? Okay, great. Maybe there is, maybe there isn't. Let's go into the parietal lobes and try to put this somewhere. And that's going to happen up here in these parietal association areas. Brodmann areas 5 and 7. Again, don't worry about the numbers. They are handy for us remembering them, but don't sweat it. No, it's the parietal lobes. And it's the part of the parietal lobes that's kind of close to the occipital lobes. That makes sense. I'm trying to say where a thing is. From here, we're going to then go talk to motor areas, right? The primary motor cortex, the premotor cortex, the frontal eye fields. And why is that? That's so we can look at a thing. If I know that the thing is a little to the right of me, I should probably have a rightward saccade so I can look at it. If I'm doing some kind of, gaze fixation, I'll talk down here to the temporal parietal occipital junction and get some smooth pursuit going on. But that's the job of the dorsal stream, to say where it is and then to help us move. Part of it might be to guide our hand, reaching out and grabbing something. It's hard to grab a cup if you don't know where it is. So we go through the dorsal stream, we say, okay, my cup is to the right. Let me prime some hand movements, some eye movements. It does help to look at a thing, to grab it. So you look at it, you reach out, you grab it, and next thing you know, you're holding your cup. Now, you don't know that it's a cup using the dorsal pathway. You know where it is. To know what it is, you got to go down here to the temporal lobes. And this is another feature of the cortex, parallel processing. At the same time, I'm doing the what is it and where is it. So these aren't happening sequentially. They're happening right at the same time. Now, that dorsal stream is kind of nifty because we can localize objects even without being able to consciously see them. So here's a gentleman who has cortical blindness. He had damage in his primary visual cortex, but those association cortices, remember, they get input from the pulvinar, and the pulvinar gets input from the superior colliculus. And so even though this gentleman can't see anything, he has no idea that what he's looking at, he can't see it, but his dorsal stream is still picking it up, and he can avoid these objects in the hallway, even though he's absolutely blind. Now, that doesn't happen if we have like retinal degeneration. Okay, this only happens with cortical blindness, if we can just damage the primary cortex but not those association cortices. So that's incredibly rare. Okay, another kind of sensation that's pretty darn important is somatosensation. And again, there's a map and it makes sense. We've got different cortical columns for all the digits and the upper limb and the trunk and the foot and the face, and you see all that. There's a somatotopic map. There's a lot of overlap, and we do indeed use lateral inhibition to focus it. So when we say that we have a digit 1 or digit 2 neuron, that just means it mostly responds to digit 1 and digit 2. But, if we were to lose a digit, we got some flexibility. And that's what's going on here. So we got the maps for digits 1, 2, 3, 4, and 5. If you lop off digit 3, which is what they did here in this poor little monkey, follow it 62 days later and construct the map. There's digit 1, 2, 4, and 5. No digit 3 because it's gone. But notice there's not just this like blank area where we don't respond to anything. No, the map for digit 2 goes right up to the map for digit 4. We've remapped the cortex because we no longer get digit 3 input. So there's nothing to inhibit the weak digit 2 and digit 4 input. So that's part of what's going on. We lose that lateral inhibition. So digits 2 and 4 can now, spread on over into the old digit 3 land. And then those synapses get used. The neurons fire together, they wire together, and we get plasticity. So in the short term, it's just some loss of lateral inhibition, but in the long term, we sprout more synapses and we make them stronger, and now we have more stable rewiring of the cortex. So there's a map. It can change. The same thing happens with vision too. Okay, so primary somatosensory cortex, we're not getting input from the lateral geniculate nucleus, no, from the ventral posterior nucleus. This will be somatosensory input from that, spinal thalamic and posterior column, medial lemniscus pathway. From the ventral posterior nucleus, we go on to the primary somatosensory cortex, which has Brodmann's areas 3, 1, 2. That's easy to remember if you're familiar with Chicago area codes. And that's kind of the order that we go through. We're going to arrive in 3, and then that'll talk to 1 or 2, and then we go to some association area. Okay, so who is S1 talking to besides the ventral posterior nucleus? Well, the other primary somatosensory cortex, so the two sides of the body know what's going on. It talks to the motor cortex, right? It tells the motor cortex, what am I feeling? That way the motor cortex knows whether or not a movement took place, for example. If we plan on bending our elbow, right, I guess flexing our elbow, then we should have an accompanying change in the somatosensory cortex if it's successful. Great. And the motor cortex can give the somatosensory cortex a heads up. Notice all these connections are reciprocal. So it can say, hey, I'm planning on flexing my elbow. The somatosensory cortex can give feedback and say, mission accomplished. This is probably how we create motor maps. I mean, think about it. The motor cortex does not talk to any muscles, right? It talks to lower motor neurons. So it's living in the dark. How does it make a map? Probably this right here. Okay, so then we have this kind of unidirectional flow of traffic out of the primary somatosensory cortex, S1, into S2, the association cortex. Hey, areas 5 and 7. That's part of the dorsal stream. So this is where we're getting some somatosensory input to help us place objects. Also useful, right, for helping guide us during reaching tasks. We got to know where it is relative to us. And we can also discriminate objects by touch, right? We can kind of get an idea of the shape of an object. Here, this person can tell it's a key because of how it feels. Part of that's the texture, part of it's the shape. So we'll put texture and shape together over here in some association cortex, and they'll say, yeah, it's a key. So the general flow through S1, again, is 3 to 1 to 2. And what you should notice here, as we're moving through, is that the receptive field gets larger. I'm just responsive to this one digit. Move over here to area 2 instead of 1. Hey, I'm responsive to 2 digits. Move into the association cortex. I'm responsive to all 5 digits. So as we go through S1 and even outside of it, receptive field size increases. And this is true for all sensory cortices. So let's say we want to distinguish texture. The general flow, we want to use cutaneous receptors. We'll obviously get input from the thalamus to area 3. Then we go to area 1 and we send that on to S2. For the shape, we don't listen to cutaneous receptors. No, instead, I care about what's the angle of my joints. I want those deep receptors, the tendon, right? Golgi tendon organs, muscle spindles. Again, they arrive at layer 3. Here not layer 3, area 3. We skip on over 1 to go to 2, and that's what goes to the association cortex. So again, parallel streams. Not what is it and where is it, but how does it feel and what's the general shape of it? put those two things together and we can identify the object. So just a quick summary, little review here. We have this sort of bottom-up processing. We get bitsy details from the afferents and we decode those in primary cortices. The information in the primary cortices gets funneled up into association cortices for us to actually make a perception. And that's how we think of sensation and perception, no matter what it is, whether it's vision, somatosensation, sound. Sound is the same, right? What are the frequencies that I'm hearing? What's the order of that? Okay, based on that, what letter sound am I hearing? Okay, I hear these letters in this order. What word is it? Just building on smaller, simpler information. Motor control, we tend to think of going the opposite way. We start with a big idea in an association cortex, and then we go to a primary cortex, and that goes and talks to the efferents. Now, in the primary motor cortex, we got our upper motor neurons that stimulate lower motor neurons. Upper motor neurons deal with movements, not muscles. Lower motor neurons in the spinal cord and brainstem deal with muscles. Okay, but who controls upper motor neurons? That's the motor association areas. So in the front. So here's my primary motor cortex, and then I've got my motor association areas, the supplementary motor cortex and the premotor cortex. And these are going to plan what actions I should carry out based on my internal plan and also based off of sensory input. If I'm going to reach for my cup, I need to know where it is. If I'm going to drive my car successfully, I need to know the color of the light. That's going to be associated in the motor association areas. They come up with a plan, they tell the upper motor neurons tell the lower motor neurons. So here's my general flow of information. Now, it's not that simple. Motor association areas can and do indeed contain upper motor neurons themselves. But again, high to low. Okay, cortical areas to know, there are 3 of them. There's M1, the primary motor cortex. I'm getting input from, well, the other M1. I'm getting input from the somatosensory cortex, so I know where my body is. I'm, of course, getting input from the supplemental motor area and the premotor cortex. That should be on here too, of course. I'm getting input from my motor association areas. I'm also, of course, getting input from the cerebellum, so I have an idea of which upper motor neurons to activate in which order, and, of course, the basal ganglia via the ventral lateral nucleus. Now, the association areas, we have the premotor cortex, which is probably larger than the primary motor cortex. This is important for externally cued movements. When we see the light turn green, we then mash the gas. And of course, these talk to each other, right? So contralateral connections, both hemispheres are talking. I'm listening to the thalamus. I'm listening to the prefrontal cortex, which is where I have my major plan. I want to get home. Okay, how do I do that? I successfully navigate the lights. And I'm also getting input from that dorsal stream. I got to know where stuff is. Who am I talking to? Primary motor cortex. And of course, all those spinal tracts. Oh, by the way, reticulospinal is in all of these. Cortical spinal tract in all of these. Okay, supplementary motor area, internally cued movements. I'm going to talk back to the prefrontal cortex. I'm getting my input from the prefrontal cortex. That's my master plan. Okay, what's my big plan? Get home. That's the thing that's predominantly driving me here. I'm still listening to the thalamus, but that's pretty much what I'm listening to is my plan. Based on my plan, I go tell the primary motor cortex or some lower motor neurons to do their thing. Okay, so M1, let's think of this as my motor neurons that are making the movement. They determine the actual direction of movement and how quickly we do it. What we do is guided by sensory input via the premotor cortex or our desired plans via the supplementary motor area. So here we're looking at recordings from the primary motor cortex when a monkey moves a lever in different directions. So there's a tone to tell it to move, and then there's the movement. And what you'll notice, boy, we get an awful lot of activity when it moves negative 90 degrees. And we get almost no activity when it moves positive 90 degrees. And as we move closer to negative 90 degrees, we get more activity. And whenever that movement is further away from negative 90, we see less activity. So just like we have a neuron for every angle of the line, we have a neuron for every direction of movement in our primary motor cortex. So little bitsy details. Okay, the premotor cortex is there to hold on to some movement plan until we get some sort of external cue. In this case, a light. So they give a light here to say, get ready to move. Some bar to the left. Again, we're dealing with monkeys here. And they've learned, when I see that light, I need to get ready to move to the left. So that's what instruction left means. And then they wait some period of time until they deliver a tone. Now, in some cases, they've delivered it early. In some cases, kind of middle of the road. And then over here, this is delivered much later. But once they deliver the tone, then they get the movement. And notice what happens. The premotor cortex is silent. But before then, it's holding on to the idea. Get ready to move left, get ready to move left, get ready to move left, get ready to move left. Then there's the tone, and they move. And then they can think about something else. As the tone takes longer, notice they're holding the activity for longer. Once they get the tone, they carry out the movement, and they move on to something else. So, our oversimplification here. Premotor cortex holds on to motor plans that it's going to tell the primary motor cortex once it gets some sort of external input, some sensory input. What about my supplementary motor area? That's all based off of internal input. So what they're doing here, they're having monkeys learn the game Simon, and that's just learning to hit these colored lights in a certain order. Now, anytime they're active, whether they're looking at the lights flashing or they're remembering the order flashing, whether it's, again, visually guided, if you see a light, you touch the button, or can you remember the order of the buttons? It involves the primary motor cortex. Of course, that's where my upper motor neurons live. For the premotor cortex, largely it's only active whenever we're following the lights. Once we learn the order of things, we don't need it anymore. Who do we need? The supplementary motor area. That's the one that remembers, oh yeah, it's the top one and then the right one and the left one and blah, 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 blah. Yeah, it's kind of active when we see the lights, but it's most active when our motor output is based off of internal input, that is memories. So, all movements require primary motor cortex. Externally guided movements are more reliant on this premotor cortex, and internally guided movements are more on the supplementary motor area. So two association areas talking to the primary motor cortex. And then those upper motor neurons go and talk to lower motor neurons. Again, we're getting from bigger ideas to smaller ideas. We're going to map from movements to muscles. Upper motor neurons are more about movements of a body part. Lower motor neurons are specific muscles. And because movements of a body part can require multiple muscles, we see that one individual upper motor neuron can control a few different pools of lower motor neurons. As we've talked about, if I want to flex my shoulder, more than one muscle group contributes to that. And therefore, all those shoulder flexing upper motor neurons have to talk to a variety of lower motor neurons. So I can stimulate my deltoid and whatever an anterior serratus is that you've told me about, I don't know. But a bunch of muscles, you know them, I don't. That's the divergence. I'm a shoulder flexing upper motor neuron. I need to talk to multiple different lower motor neurons. I need to diverge. Now there's also convergence. That deltoid is involved with more than just shoulder flexion. It might also be, abduction or something along those lines. Again, you know more than I do. So we'll see multiple upper motor neurons activated for the same movement because they converge on the same lower motor neurons here. And that convergence, right, this overlap here is a whole lot more extensive for muscles than movements. And this is again showing you upper motor neurons a little more specific for movements, than they are for muscles. Okay, here we just have them move the shoulder. We see a lot of activity here in these regions of the primary motor cortex. This is more like the proximal, and then the distal muscles are over here in the kind of the yellow. When we have them move their wrist around, these are the upper motor neurons that are active. And notice, not a ton of overlap between wrist and shoulder. But whenever we look at whether or not a certain muscle is active, for example, the deltoid, or whatever the hell this is, I'm guessing it's some kind of extensor muscle for the fingers, maybe controlled by the radial nerve. I don't know. You tell me. But notice tons of overlap down here when we look at how an upper motor neuron maps to muscles. It's in no way specific. What motor neurons map to the deltoid? Tons of them. Tons of upper motor neurons map to the deltoid, because movement of your arm or your hand can involve the deltoid, right? If you're thinking of moving your hand forward, that may very well involve shoulder flexion, because we're not thinking about body parts. We're not thinking about muscles. We're thinking about moving a body part. I want to move my hand forward. I'm going to accomplish that using the elbow and the shoulder. If I think of doing whatever this thing does with I'm assuming fingers, well, lots of upper motor neurons might want to do that, right? Even though this one right over here should just map to, mostly the shoulder. I don't know, maybe I'm reaching out, and part of that's extending the fingers, assuming this thing does that. Again, I have no idea what it does. But we get these kind of different orders of things. We think of sensation and perception as being bottom up, and then motor systems as being top down. Now, I do want to say that perceptions, right, sensation and perception isn't only bottom up, it's also top down, right? So this is also true in sensory systems. The same thing we saw in motor, true over here. This is what we call the McGurk effect. This is a wonderful illustration of it, and hopefully it works. Let's give her a shot.
Ba 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 ba.
Okay, have a look at this guy.
Ba 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 ba.
I hear ba ba.
Ba 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 ba
Here.
ba ba. Ba 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 ba.
I hear the when I'm looking here, but I look here.
Ba 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 ba.
Va va.
Ba 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 ba.
So it's either ba, the, or va. Which one is it? It's none of them. It's just, I think it might just be da. But depending on where you're looking, the visual input is going to change what you hear.
Ba ba.
Go ahead, close your eyes.
Ba ba.
It's the same
Ba
sound.
ba.
The only difference is what visual input you're giving. And so that's showing us the top-down processing. Now, make sure you can go through your two different streams. Your ventral stream, your dorsal stream, going from the visual cortex to the temporal lobes. What is it? Parietal lobes. Where is it? Right? Make sure we get some idea about why the motor and somatosensory cortices would want to talk to each other. That's how we make a map. We can give the somatosensory cortex a heads up. We can tell the motor cortex that a movement actually took place, right? Know what's going on with areas 5 and 7. Hey, they're in the dorsal stream, helping us know where an object is. Oh, they're also getting input from that somatosensory cortex, so we can feel and determine the shape of an object. And they're providing input to the premotor cortex, right? So we can visually guide our movements. And remember, premotor cortex, there we're thinking, externally guided stuff, internally guided, more supplemental motor area. And both of these then influence our actions by projecting to the primary motor cortex, which then controls lower motor neurons. Yes, these also do it too, I know. But let's accept our kind of simplified version here, where our association areas guide upper motor neurons using kind of distinct inputs. All right, now if there's any distinct input you wanna ask a question about, put some distinct inputs into the questions box, and I'll make sure we talk about them in class. I'll see you later.